Welcome to this video where I will be explaining the cellular level of organization. Remember to like, share, and subscribe to my YouTube channel for more content. About 200 different types of specialized cells carry out a multitude of functions that help each system contribute to the homeostasis of the entire body. At the same time, all cells share key structures and functions that support their intense activity. The cytoplasm consists of all the cellular contents between the plasma membrane and the nucleus. This compartment has two components, cytosol and organelles. Cytosol, the fluid portion of cytoplasm, contains water, dissolved solutes, and suspended particles. Surrounded by cytosol are several different types of organelles. Each type of organelle has a characteristic shape and specific functions. Examples include the cytoskeleton, ribosomes, endoplasmic reticulum, Golgi complex, lysosomes, peroxisomes, and mitochondria. The nucleus is a large organelle that houses most of a cell's DNA. Within the nucleus, each chromosome, a single molecule of DNA associated with several proteins, contains thousands of hereditary units called genes that control most aspects of cellular structure and function. The plasma membrane, a flexible yet sturdy barrier that surrounds and contains the cytoplasm of a cell, is best described by using a structural model called the fluid mosaic model. According to this model, the molecular arrangement of the plasma membrane resembles an ever-moving sea of fluid lipids that contains a mosaic of many different proteins. Some proteins float freely like icebergs in the lipid sea, whereas others are anchored at specific locations like boats at a dock. The membrane lipids allow passage of several types of lipid-soluble molecules, but act as a barrier to the entry or exit of charged or polar substances. Some of the proteins in the plasma membrane allow movement of polar molecules and ions into and out of the cell. Other proteins can act as signal receptors or adhesion molecules. The basic structural framework of the plasma membrane is the lipid bilayer. Two back-to-back layers made up of three types of lipid molecules, phospholipids, cholesterol, and glycolipids. About 75% of the membrane lipids are phospholipids, lipids that contain phosphorus. Present in smaller amounts are cholesterol, which is a steroid with an attached hydroxyl group, and various glycolipids, which are lipids with attached carbohydrate groups. In this way, the heads face a watery fluid on either side, cytosol on the inside and extracellular fluid on the outside. The hydrophobic fatty acid tails in each half of the bilayer point toward one another, forming a nonpolar hydrophobic region in the membrane's interior. The bilayer arrangement occurs because the lipids are amphipathic molecules, which means that they have both polar and nonpolar parts. Cholesterol molecules are weakly amphipathic and are interspersed among the other lipids in both layers of the membrane. Membrane proteins are classified as integral or peripheral according to whether they are firmly embedded in the membrane. Integral proteins extend into or through the lipid bilayer among the fatty acid tails and are firmly embedded in it. Most integral proteins are transmembrane proteins, which means that they span the entire lipid bilayer and protrude into both the cytosol and extracellular fluid. As their name implies, peripheral proteins are not as firmly embedded in the membrane. They associate more loosely with the polar heads of membrane lipids or with integral proteins at the inner or outer surface of the membrane. Many membrane proteins are glycoproteins, proteins with carbohydrate groups attached to the ends that protrude into the extracellular fluid. Some integral membrane proteins form ion channels, pores or holes through which specific ions, such as potassium ions, can flow to get into or out of the cell. Other integral proteins act as carriers, selectively moving a polar substance or ion from one side of the membrane to the other. Carriers are also known as transporters. Integral proteins called receptors serve as cellular recognition sites. Each type of receptor recognizes and binds a specific type of molecule. Some integral proteins are enzymes that catalyze specific chemical reactions at the inside or outside surface of the cell. Integral proteins may also serve as linkers, which anchor proteins in the plasma membranes of neighboring cells to one another or to protein filaments inside and outside the cell. Membrane glycoproteins and glycolipids often serve as cell identity markers. They may enable a cell to recognize other cells of the same kind during tissue formation, or to recognize and respond to potentially dangerous foreign cells. The selective permeability of the plasma membrane allows a living cell to maintain different concentrations of certain substances on either side of the plasma membrane. A concentration gradient is a difference in the concentration of a chemical from one place to another, such as from the inside to the outside of the plasma membrane. Substances generally move across cellular membranes via transport processes that can be classified as passive or active, depending on whether they require cellular energy. In passive processes, a substance moves down its concentration or electrical gradient to cross the membrane using only its own kinetic energy. There is no input of energy from the cell. In active processes, cellular energy is used to drive the substance uphill against its concentration or electrical gradient. The cellular energy used is usually in the form of ATP. Another way that some substances may enter and leave cells is an active process in which tiny spherical membrane sacs referred to as vesicles are used. Examples include endocytosis, in which vesicles detach from the plasma membrane while bringing materials into a cell, and exocytosis, the merging of vesicles with the plasma membrane to release materials from the cell. Diffusion is a passive process in which the random mixing of particles in a solution occurs because of the particles' kinetic energy. Both the solutes, the dissolved substances, and the solvent, the liquid that does the dissolving, undergo diffusion. If a particular solute is present in high concentration in one area of a solution, and in low concentration in another area, solute molecules will diffuse toward the area of lower concentration. They move down their concentration gradient. Several factors influence the diffusion rate of substances across plasma membranes, including steepness of the concentration gradient, temperature, mass of the diffusing substance, surface area, and the diffusion distance. Simple diffusion is a passive process in which substances move freely through the lipid bilayer of the plasma membranes of cells without the help of membrane transport proteins. Solutes that are too polar or highly charged to move through the lipid bilayer by simple diffusion can cross the plasma membrane by a passive process called facilitated diffusion. In this process, an integral membrane protein assists a specific substance across the membrane. The integral membrane protein can be either a membrane channel or a carrier. Osmosis is a type of diffusion in which there is net movement of a solvent through a selectively permeable membrane. Like the other types of diffusion, osmosis is a passive process. In living systems, the solvent is water, which moves by osmosis across plasma membranes from an area of higher water concentration to an area of lower water concentration. Another way to understand this idea is to consider the solute concentration. In osmosis, water moves through a selectively permeable membrane from an area of lower solute concentration to an area of higher solute concentration. During osmosis, water molecules pass through a plasma membrane in two ways. One, by moving through the lipid bilayer via simple diffusion, as previously described, and two, by moving through aquaporins, integral membrane proteins that function as water channels. A solution's tonicity is a measure of the solution's ability to change the volume of cells by altering their water content. Any solution in which a cell maintains its normal shape and volume is an isotonic solution. Cells placed in isotonic solution do not alter in volume. Hypotonic solution is a solution that has a lower concentration of solutes than the cytosol. Cells placed in hypotonic solution swell. A hypertonic solution is a solution which has a higher concentration of solutes than does the cytosol. Cells placed in hypertonic solutions shrink. Active transport is considered an active process because energy is required for carrier proteins to move solutes across the membrane against a concentration gradient. Two sources of cellular energy can be used to drive active transport. 1. Energy obtained from hydrolysis of adenosine triphosphate, or ATP, is the source in primary active transport, and 2. Energy stored in an ionic concentration gradient is the source in secondary active transport. A vesicle is a small spherical sac. A variety of substances are transported in vesicles from one structure to another within cells. Receptor-mediated endocytosis is a highly selective type of endocytosis by which cells take up specific ligands. Scene 2. Phagocytosis. Phagocytosis is a form of endocytosis in which the cell engulfs large solid particles such as worn-out cells, whole bacteria, or viruses. Scene 3. Pinocytosis. Pinocytosis is a form of endocytosis in which tiny droplets of extracellular fluid are taken up. Transport in vesicles may also be used to successively move a substance into, across, and out of a cell. In this active process is called transcytosis. Scene 2. Cytoplasm. Cytoplasm consists of all the cellular contents between the plasma membrane and the nucleus, and has two components, 1, the cytosol, and 2, organelles, tiny structures that perform different functions in the cell. The cytosol, or intracellular fluid, is the fluid portion of the cytoplasm that surrounds organelles and constitutes about 55% of total cell volume. Scene 2. Cytosol composition. Although it varies in composition and consistency from one part of a cell to another, cytosol is 75 to 90% water plus various dissolved and suspended components. Among these are different types of ions, glucose amino acids, fatty acids, proteins, lipids, ATP, and waste products. Scene 2. Cytosol function. The cytosol is the site of many chemical reactions required for a cell's existence. Thank you for watching this video. Next video we will look at the structure and functions of various cell organelles, so remember to like, share and subscribe to my YouTube channel.